Hi guys, welcome to my YouTube channel. Don't forget to subscribe and share. Alright guys, so this is the last part. Okay, the last subtopic in chapter 4, biocatalysis. And the subtopic is 4.3, inhibitors. Okay, so inhibitors. Okay, so basically kalau kita compare inhibitor and cofactor, dia orang memang sangat lain lah. Okay, cofactor will help the enzyme to function more effectively, but inhibitor dia merencat. So dia boleh mengganggu kerja-kerja enzim. Right, this inhibitor is a substance that bind to an enzyme and decrease or stop the enzyme activity. See, it will decrease or stop the enzyme activity. It will bind either to the active site or allosteric site. Okay, so cofactor tadi, it bind to allosteric site. But this inhibitor, it can bind both, okay, at allosteric site or the active site of enzyme. So the rate of enzyme control reaction may be decreased by the presence of inhibitor. So in presence of cofactor, the enzyme activity is more effectively lah, bagus. But in presence of inhibitor, it will decrease or stop the enzymatic activity. Alright, we have two types of inhibitor: revisible inhibitor, irrevisible inhibitor. This revisible inhibitor has two types, competitive and non-competitive. Actually, irrevisible inhibitor has two types also with the same type, which is competitive and non-competitive. Okay? So, revisible, reverse. Okay, so reverse. Apa maksudnya? This inhibitor bind to the enzyme by weak interaction or weak bond. It bind to the enzyme by forming a weak interaction or weak bond. So it bind, the effect of this inhibitor is only temporary and it can easily be removed. Okay, so it will cause no permanent damage to enzyme. And removal of inhibitor restore the activity of enzyme to normal. Apa maksudnya? Maksudnya this inhibitor, okay, it will bind to the enzyme temporary, maksudnya dia hanya bind sementara. Okay, dan dia punya binding tu pula adalah dalam bentuk yang lemah, weak bond or weak interaction. Jadi dia senang untuk keluar masuk. So bila dia masuk, dia bind to the enzyme, dia akan decrease enzymatic reaction. Okay, dia akan decrease enzymatic reaction. But bila dia left the enzyme, bila dia keluar tinggalkan enzyme itu masih okay and the enzyme will able to conduct its function. Dia tak ada masalah sebab dia tidak berubah. The enzyme has no permanent damage. So the enzyme can continue its function. Okay, so for revisible inhibitor, we have two types. First, competitive, and second one is non-competitive. As you can remember from the first study, slide yang pertama tadi, kita dah kata, the inhibitor can bind to both active site and also allosteric site of enzyme. Okay, so for competitive inhibitor, why we call competitive? Because it binds to the active site of enzyme. It binds to the active site of enzyme. So competitive means the substrate and the inhibitor will compete to bind to the same site, which is the active site. Okay? Jadi kita kata dia competitive inhibitor. Kenapa? Sebab dia bersaing dengan substrate untuk sama-sama juga memilih to bind to the active site. Substrate and this inhibitor, they may bind to the active site. So, siapa dulu dapat, dia lah yang menang. Dia lah yang berjaya. So, to bind with the active site, this inhibitor have similar structure to the substrate. Okay, since dia nak bind pada active site, dia mesti ada similar structure to the substrate. Structure dia pun mesti sama dengan substrate, barulah dia get fit to the active site. Okay, so ini yang dipanggil sebagai competitive inhibitor. Okay, why it remain bound to the active site? Okay, it prevent the substrate molecule to occupy the active site. Means that when this inhibitor bind to the active site, it will block. So it will prevent the substrate from bind to the active site. So no enzymatic reaction occur. Okay. So here is the graph for competitive inhibitor. Okay, other substrate will bind only to unaffected enzyme. The same quantity of product is formed, but it takes longer to make the product. The rate of reaction is reduced. Okay, so here we have two curves. The green one show the curve without inhibitor, and the red one show the reaction with competitive inhibitor. Without inhibitor, the rate of reaction is higher, okay, until the active site is fully saturated lah, okay, with the substrate. But for the case in the presence of competitive inhibitor, the rate of reaction is a bit lower. Lebih rendah. Okay, kenapa lebih rendah? Because we know that after the inhibitor bind to the active site, it will block the substrate from bind to the active site. So no enzymatic reaction occur. So that's why the rate of reaction decrease. But once the inhibitor left the active site, then the substrate bind to the active site so that the reaction occur, and at the end, the same amount of product may form. But it will take a long period of time compared to the condition with no inhibitor. Okay. The greater concentration of substrate, the greater their chance of finding active site. So how kita nak overcome? Macam mana kita nak overcome? Nak bagi supaya yang menang untuk fit dengan, untuk attach dengan active site tu adalah substrate? Mudah saja. By increase concentration of the substrate. When the concentration of substrate increase, it will increase the chance of the substrate to bind to the active site. Okay, compared to the inhibitor. Example is succinic acid. Okay, succinic acid is the substrate, and then the malonic acid is the inhibitor. The enzyme is succinic dehydrogenase, this brown one. Okay, so look at the shape of the active site. Okay, and the shape of active site is compatible to both for the succinic acid, their substrate, and also to the malonic acid, which is the inhibitor. So in this case, malonic acid is competitive inhibitor. Okay, inhibitor absence, the substrate attached to the active site of enzyme. So the reaction occur to produce fumaric acid, which is the product. But when the competitive inhibitor present, which is the malonic acid, what happened? The inhibitor prevent the normal enzyme substrate complex being formed, and the reaction rate is reduced. So the presence of malonic acid, the competitive inhibitor will reduce the rate of reaction. Because in this is not called as enzyme substrate complex. So since no enzyme substrate complex, no reaction occur, no product will form. The product will form when this malonic acid left the active site, so that the succinic acid bind to the active site, the reaction can form, and the product may produce. Next is non-competitive inhibitor. Non-competitive inhibitor means this inhibitor will not compete to the substrate for the active site. Dia tak akan bersaing pada active site. Tapi dia akan duduk dekat mana? It will bind to the allosteric site. Okay, this type of inhibitor will bind to the enzyme at allosteric site. So what is allosteric site? The specific receptor site on some part of enzyme molecule other than active site. So the binding alter the shape of the active site of the enzyme. So active site cannot bind to the substrate. Nampak ke tidak? For this non-competitive inhibitor, yes, it not bind to the active site. It bind to allosteric site. But the binding of this non-competitive inhibitor will alter the shape of active site. So the shape of active site will not compatible with the shape of substrate. So still the substrate cannot bind to the enzyme, so no reaction occur and no product will form. So what happened to the rate of reaction? The rate of reaction reduced. Okay, so example here, okay, this is the substrate and here is the enzyme. Yes, the substrate is exactly compatible with the active site of enzyme. Okay, and here is the allosteric site. Okay, when this non-competitive inhibitor bind to the enzyme, it will cause the change of shape, change conformational shape of the active site. So the active site of enzyme change cause this substrate cannot bind to the active site. Nampak tak? Dia tak boleh nak bind. Sama juga dalam kes ini. When the presence of non-competitive inhibitor to the allosteric site, and then this active site change conformation, it will not compatible with the substrate anymore. So the substrate cannot bind to the active site. No reaction occur. So the rate of reaction reduce. Okay. Here is the graph, okay, to show non-competitive inhibitor. Okay, this straight line refer to reaction without non-competitive inhibitor. Okay, so the rate of reaction is high, and then the product form is also in high rate. But in presence of non-competitive inhibitor, what happen is, okay, it will reduce the rate of reaction, and at the end, what happen? The product is lower than yang sepatutnya. Okay, kenapa? Because in this case, the increase of substrate will not reduce the effect of inhibitor. The increase of the substrate will not reduce the effect of inhibitor. Kalau kita tambah substrate banyak mana pun, dia tak akan menyebabkan substrate boleh duduk dekat active site. Tak. Sebab apa? They are not compete. Inhibitor will bind to allosteric site, while the substrate, okay, bind to active site. But then, since active site sudah berubah, so substrate tak boleh nak bind. Bila substrate tak boleh nak bind, memang tak akan ada reaction. Okay, takkan ada reaction yang berlaku. Jadi rate of reaction rendah. And then the amount of product form pun akan rendah. Okay, here is the example lah. Okay, example is isoleucine. Okay, the product, the substrate is threonine. Enzyme is threonine deaminase, and the product is isoleucine lah. Okay, so apa yang jadi dengan isoleucine ni? Okay, the isoleucine ni dia boleh jadi non-competitive inhibitor. Macam mana? Okay, kalau kita tengok dekat sini, okay, the product, the substrate is threonine. Threonine bind to the enzyme which is threonine deaminase and produce isoleucine. So this isoleucine, dia boleh bind pada allosteric site of threonine. So they bind to the allosteric site of threonine. So when it bind to allosteric site of threonine deaminase, bind to the allosteric site of the enzyme, which is threonine deaminase, the active site of enzyme change, and the threonine cannot bind to the enzyme. Okay. Then what happened? Of course, no reaction and no product may form. Okay. So the binding of allosteric site, as isoleucine accumulate, it will slow down its own synthesis. By binding to allosteric site of enzyme, the pathway is switched off. Okay, boleh faham? Isoleucine ni adalah produk. By the same time, dia juga act as non-competitive inhibitor. Alright, here is the summary. Okay, the revisible inhibition of enzyme activity. Here is the substrate, here is the active site. So we know substrate bind to the active site. But in presence of competitive inhibitor, okay, this competitive inhibitor bind to the active site. So it will prevent the substrate from bind to the active site. Okay, and the second type is non-competitive inhibitor, which this inhibitor will bind to the allosteric site. And the binding of the inhibitor, it will change the conformation of the active site. So this substrate cannot bind to the active site. So for these two, competitive inhibitor or non-competitive inhibitor, it may reduce the rate of enzyme reaction. Okay. So the second types of inhibitor, okay, so again, the competitive versus non-competitive inhibitor, okay. For competitive inhibitor, remember, it has the same structure, the same shape, okay, as the substrate, so that dia boleh bind dekat active site. Okay, but for non-competitive inhibitor, it has different structure from the substrate, but it's okay because it will bind to the allosteric site. The binding of this non-competitive inhibitor to the allosteric site change the conformation of active site, prevent the substrate from bind to the active site. Okay, here is the graph to show the rate of enzymatic reaction with and without competitive inhibitor. Okay, so also with non-competitive inhibitor. So we have three curve here: rate of reaction, substrate concentration. Okay, for the first curve, the purple color, it show normal enzyme, means without any inhibitor. So the rate of reaction is high. Okay. And then here is the amount of product lah. Okay, because we know when the substrate concentration increase until a certain period of time, when the active site of enzyme is fully saturated, so what happened to the rate of reaction? They may not increase anymore. Okay, second curve, it show competitive inhibitor. In competitive inhibitor, the rate of reaction decrease. Okay, from decrease lah sikit. Tapi apa yang berlaku, the product tetap sama. Kerana apa? Okay, because when we increase the substrate concentration, okay, it will increase the chance of substrate to bind with the active site compared to the inhibitor. Boleh faham? In competitive inhibitor, the rate of reaction memang rendah berbanding yang normal, tetapi at the end produknya tetap sama. Kerana apa? Because when increase the substrate concentration, it will increase the chance of substrate to bind to the active site compared to the inhibitor. So at the end, tetap juga akan hasilkan the amount of product yang sama. Okay, the third curve is refer to non-competitive inhibitor. The rate of reaction is lower. Yang paling rendah, lowest. Okay, and what happen is, this non-competitive inhibitor, when we increase the substrate concentration, it will not increase the rate of reaction anymore. Dan dia tidak akan dapatkan produk yang sama banyak. Kerana apa? Sebabnya substrate concentration will not affect the apa kita panggil, dia tak akan memberi kesan kepada non-competitive inhibitor. Kerana apa? Non-competitive inhibitor will bind to allosteric site. So even though we increase the substrate concentration, since the non-competitive inhibitor bind to the allosteric site, substrate memang tak boleh nak bind. Okay, tambahlah banyak mana substrate concentration sekalipun, sebab they are not compete to the same active site. Okay, so last, okay, for inhibitor is irrevisible inhibitor. Revisible, ingat tadi the revisible inhibitor, the effect is a short period ataupun kita panggil temporary. Sebab apa? Sebab dia bind secara loosely and then temporarily bila dia keluar, so it will not cause permanent damage to enzyme. So enzyme can be reusable. Okay, but for irrevisible inhibitor, tengok ni it bind to the enzyme by forming covalent bond. So bila kata dia bind dengan covalent bond, mean it bind tightly and permanently. Dia bind tightly and permanently to the enzyme. So bila dia buat macam tu, dia bind tightly and permanently, it will cause permanently inactive enzyme. Dia akan menyebabkan permanently inactive enzyme. So bila kata permanently inactive enzyme, maksudnya dia dah jadi irrevisible. Enzim tadi dah tak boleh nak balik pada bentuk asal. Okay, boleh faham konsep tu? Irrevisible inhibitor means inhibitor tersebut bind secara tightly and permanently to the enzyme and cause the enzyme inactive, change its conformational shape, okay, permanently. So permanently lah dia akan dalam keadaan inactive. So resulting the enzyme will permanent lost its catabolic properties. Maksudnya enzyme tu dah tak boleh langsung digunakan. Okay. So example here is the active site, here is irrevisible inhibitor. When irrevisible inhibitor bind, okay, apa yang dia buat? The enzyme function is permanently disabled. Because of what? It will bind permanently to the active site. So permanently substrate cannot bind to the active site. Okay, dia bind secara permanent. Bila dia bind secara permanent, apa jadi? Substrate memang tak boleh nak bind selamanya. Okay, so the enzyme function is permanently disabled. So here is the differences between irrevisible inhibitor and revisible inhibitor. For irrevisible, it will bind, okay, and then cause permanently bind to the enzyme. So substrate will permanently cannot bind to the enzyme. Memang tak boleh nak bind selamanya. But untuk revisible inhibitor, once inhibitor bind to the enzyme, okay, the rate of reaction reduce ataupun this enzyme memang tak boleh tak boleh carry out its catabolic function lah. Tetapi bila dia keluar, bila revisible inhibitor left the enzyme, tak kisahlah left the active site or left the allosteric site according to the types, sama ada dia kompetitif ataupun non-kompetitif, bila dia keluar saja daripada enzyme, apa yang jadi? The substrate can bind to the enzyme. Okay. Example of irrevisible inhibitor is cyanide. Okay, so the cyanide is a chemical poison that inhibit the normal activity of enzyme. The enzyme is cytochrome C oxidase in electron transport chain in mitochondria. So this cyanide be tightly bound to the iron atom of the enzyme, making it unable to transport electron. So dia akan menyebabkan the enzyme cytochrome C oxidase tak boleh nak transport electron. So therefore, passage of electron to oxygen in electron transport chain is blocked. ATP production is stopped. So nampak, this cyanide, dia bind to enzyme cytochrome C oxidase. So the cytochrome C oxidase cannot conduct its function. So what happened? It will cause the passage of electron to oxygen in ETC being blocked. So ATP production is stopped. So consequence dia sangat besar. Okay, here is the example that you can read. Okay, this one pun sama jugalah. The paraquat, nerve gas. So jenis irrevisible heavy metal pun yes. Okay, for example mercury. So that's why it's very dangerous bila ada mercury yang leaking. Contohnya kamu nak main thermometer, tiba-tiba thermometer kamu pecah. So kamu memang dilarang sepenuhnya pegang mercury tu. Kenapa? Because mercury ni adalah heavy metal dan dia act as irrevisible inhibitor. So when dia masuk dalam badan kita, bayangkan dia masuk dalam badan kita, dia bind pada enzyme, enzyme dalam badan kita akan permanently damage. So enzyme itu langsung nanti tak boleh nak carry out its function. Jadi apa jadi dengan metabolic activity dalam badan kita? Akan terganggu dan apa yang boleh berlaku? Boleh maut. Okay, so that's all for chapter 4. Okay, let's have a quick revision untuk subtopik ini.